What's up? All right, are we ready to go? An overwhelming yes is what I just heard. We're excited and we're ready to go. Perfect. Perfect. All right. Did we all do better on test 2 than test 1?
Yes.
Yes? Okay, good. Like significantly better or 1%? Okay.
16%. I'm not even kidding. That was
Okay,
great.
that's good. That's good. So,
Good job.
all right. So that test's pretty straightforward, right? Not nearly as tricky as test 1. I still got to work on making my test 1 tricky.
Is test 3 pretty straightforward?
I think test 3 is pretty straightforward as well. So, yeah, there's not really as much that he can trip you up. Like we'll see with chapter 11, really straightforward. Just got to remember a few different enzyme names, and we're good to go.
Like the best time of
But all right, so do we have any questions from the exam? Was there any that stuck out to y'all? I know that was last week, but are we just like wiping all that already from our memory? Okay. Thanks for being here after muscles. Appreciate it. For those of you watching at home, thanks for joining eventually.
I just saw her. Oh, yeah, but no one was in there, so I was waiting to play.
How far have y'all gotten in lecture thus far? Have y'all finished chapter 11? Have y'all?
Yeah. Well, I would know.
Are y'all on?
Started on chapter 12.
You started chapter 12. All right. So we're just going to do chapter 11, make it really easy today. Y'all are fried. Y'all don't want to stay here for 2 hours. So cool. We got to go get Parker West action going.
No, we already did that.
Again, times 2.
Yep.
Y'all took 2 muscle tests, right? So we got to go twice. Anyway, all right. So chapter 11, oxidative phosphorylation. Who can tell me where this happens at? Where in the mitochondria?
No, that's like
Mitochondria's right. Is it the matrix, right? Yeah, mitochondrial matrix. All right, so we started, so glycolysis was cytosol, TCA cycle was the outer membrane, right? So now we're going that next step into the matrix, inner membrane. So what we did with the TCA cycle was we made the GTP, but mainly we made the 3-NADH and the FADH2, yeah? So now we're going to take those hydrogens that we took off of I guess throughout the entire cycle, and we're going to take those hydrogens off and use them to make ATP. So this is where we get the big amounts of ATP being made, and that's where our biggest gains are at. Gains, hashtag gains, like we said however many weeks ago. All right. So this is the last pathway in the cycle. So glycolysis, TCA cycle, oxidative phosphorylation. So oxidative phosphorylation, what have we seen that's similar to that up until this point?
oxidative phosphorylation, is that what you said?
Yeah, what have we seen similar to that? It's the type of reaction for alpha ketoglutarate dehydrogenase and isocitrate. Yeah, so we've seen an oxidative decarboxylation, and now we're just seeing oxidative phosphorylation. So an oxidizing type of reaction that is going to be involving putting on phosphate groups onto ADP to make ATP. So that's kind of the overall structure, but it's the final pathway, and it's in all the aerobic organisms. So electrons derived from the reducing agents, so NADH, FADH2, from the various pathways are flown through a series of membrane-bound electron carriers. We'll get to there. Really, the whole thing is releasing protons, making a proton gradient. So kind of like how in the sodium-potassium pump, where we make that gradient of sodium outside the cell and potassium inside the cell, it's the same thing. We're putting a bunch of hydrogens outside the cell, making that more positive, and then going down that gradient is going to be making the ATP. And that's really all it is, with just a couple steps in between. All right, so glucose is completely oxidized to CO2 through glycolysis and citric acid cycle. We've already made 2 of those. We talked about that. Removes electrons, electron transfer, connects to ATP. Just said it. All right. So glucose oxidation yields electrons. There's a couple important points on this page. So we have this overall equation of glucose plus oxygen yields carbon dioxide plus water. And really that's broken up into 2 different steps, so 2 half reactions. The important thing to keep in note here is that there is no direct transfer of electrons to O2. Easy test question. So just keeping in mind that we're not directly transferring any pairs of electrons onto oxygen by itself. So the first half reaction is we're taking glucose and we're adding water and we're making CO2, hydrogens, and electrons. The second half of that is we're taking oxygen, we're adding those hydrogens and electrons to it to make water. So we're adding the hydrogens, electrons to oxygen to make water, but not directly. It's all indirectly, using these carriers, NADH, FADH2, which will become more apparent here in 2 pages. And dehydrogenases are what generate those molecules. We've got a nice little paper here telling you where exactly, so if y'all had looked to this page before the last test, this would have told you where all the enzymes are that produce NADH or FADH2. But we already know it's in our memory now, right? Do I need to go over, I went over this a couple weeks ago. Do I need to go over it one more time of how to make NADH, or are we good? We'll do it again.
Yeah, I remember, but is it like directly question it on the test?
Probably not.
Are you just talking about that reaction down there?
Just how we go, how NADH gets its hydrogens. We'll just do it. We'll just be quick about it. All right, so hydrogen. It's 2 hydrogens. It's typically as H2. It's more stable than just H by itself. So if it's H2, is that polar or nonpolar? Covalent bond. Nonpolar, right? Because they're the same. So there's no polarity within it. All right, so if we break it apart, we can have either a hydride ion. So either one hydrogen gets both electrons, and one hydrogen will not get any electrons. So one will have a negative charge, one will have a positive charge. So now we've got this NAD plus saying, hey, I'm positively charged. Hey, hydride, you're negatively charged. Let's combine. So then you get NADH plus that free-floating H, so which is why a lot of times you'll see NADH plus H, just because there were those 2 hydrogens in the first place. Because like H2O, right? So if we took the oxygen off, it's going to leave those 2 hydrogens. Cool. All right, so the next couple pages are going to be about shuttle systems. Did y'all have a decent feel for these? Just be honest with me. Do y'all have like any idea what any of this stuff is?
At all, because we were still going, like, we hadn't taken the test yet.
Oh, you did this before the test?
Yeah. Like,
I did really
so-
well on the test. I have no idea what's on this.
Yeah, we did.
Perfect. Okay, so, all right, so we made NADH in the cytosol, yeah?
Yeah.
What, because of glycolysis, it was what, G3P dehydrogenase? Glycero-phosphate 3-phosphate dehydrogenase is what made it. So since oxidative phosphorylation happens inside that mitochondrial membrane, on that inner membrane, or in the, whatever the other word was, matrix, we need to get it in there somehow. We need to get that hydrogen or those electrons in there. And so really all this shuttle system is talking about is saying, hey, we've got this hydrogen outside of the mitochondria, and these things are what's going to bring it inside. Really simplified version, that's all it is. So there's 2 different ones. We've got the malate-aspartate shuttle, and we've got the glycero-phosphate shuttle. But the first one, we're going to start with the malate-aspartate shuttle first. We'll go to the picture, but really quickly, quick note, that malate-aspartate shuttle just kind of tells you where they are. He might ask you where these are located at. So functions, the malate-aspartate is mostly in the liver, kidney, and heart mitochondria, whereas the glycero-phosphate shuttle are mostly in the skeletal muscle and brain.
Is that supposed to be like the mitochondria, like liver mitochondria, kidney mitochondria?
Yes. So, all right, so malate-aspartate shuttle. I like using the picture first, and then we can go through all the verbiage, just because verbiage is difficult sometimes. But, all right, so main things. It's the malate-aspartate shuttle. So those are going to be the two things that are going through the membrane. That's why it's named. Makes a lot of sense. So malate goes through the membrane. Aspartate goes through the membrane. But what we're starting with is oxaloacetate. And if we can get all of those, just saying, hey, in the middle we've got oxaloacetate, at the top we've got malate, bottom we've got aspartate. That's really just the big bulk of it. So we're going to start with oxaloacetate, and I'm going to give you all a little analogy. It took me a while to figure out to make it like me come out on top. So we've got tri-1s, our oxaloacetate. All right. So y'all are just chilling here. Y'all can't get in the club. Y'all aren't cool enough. Y'all are just little peasant tri-1s. Y'all don't know the bouncer yet. So what y'all are going to do is we've got this NADH here from the cytosol, and y'all are going to get the hydrogens off of NADH, and you're going to turn it into malate. So now your tri-1's dressed as Andrew, and like Andrew can get in the club. So malate gets in. Yeah, I got it written down. Took me a while. So the main, I guess, so that's from the molecule side. So we've got this NADH here. NADH can't just go through by itself. It needs the shuttle. So NADH latches on. It attaches to oxaloacetate with malate dehydrogenase, produces malate. Now y'all are dressed like me or whatever. Y'all can get in. So y'all go through. So y'all go through the malate alpha-ketoglutarate carrier. So y'all are in. Now y'all don't want to be dressed like me the whole night, so y'all dump that those hydrogens and reform NADH on the inside. And y'all go back to y'all's tri-1 cells, oxaloacetate. But the whole thing, though, was NADH from the outside. Now we've got NADH on the inside. And malate dehydrogenase as well. So malate dehydrogenase is what's in charge of putting the hydrogen onto oxaloacetate to make malate. And it's also used to turn malate back into oxaloacetate. So really simple. You only have to remember one enzyme that deals with it. Because it's just talking about taking a hydrogen off of NADH and putting it onto oxaloacetate over here. But on this side, it's saying we're taking the hydrogen off of malate and putting it onto NAD over on this side. Really simple. So we've accomplished our goal, right? We've accomplished our goal. This is where the analogy breaks apart, and I didn't have any time. I was in class. I had to learn myself. So we've got oxaloacetate here. We need to go. Oxaloacetate needs to get back on the outside so that it can repeat the process. Yeah, so that it can keep getting in the club. So at the end of the night, you got to go home. Next day, go out and do it again, what have you. So, but oxaloacetate, we already said, can't just diffuse straight through. So it's not going to turn back into malate, because if it did that, it would take away the hydrogen, yeah? It would put that hydrogen back onto it and defeat the whole purpose. So what it does is there's a transamination reaction with glutamate, and it produces aspartate. So the second half of the shuttle is the malate-aspartate. So oxaloacetate turns into aspartate and can go through where it goes through another transamination reaction to turn back into oxaloacetate. We're back where we started. There's a few more intricacies, but overall,
We get drunk,
We got kicked out.
and then we go home, and then we're hungover, so you're still kind of drunk, and then you sober up finally, and you're back to your normal self.
perfect. Perfect. So overall, we've got NADH. We've got NADH from the cytosol. It needs to get on that side. So combines with oxaloacetate to make malate. Malate can go through the shuttle, or it can go through the carrier. Malate's on the inside. It's having a good time. Releases the hydrogens back. So we've reformed NADH on the inside. So by taking off the hydrogens, we're back to oxaloacetate. Oxaloacetate needs to get out, so it turns into aspartate. Aspartate can go through this carrier over here, and then it turns back into oxaloacetate. That's like overall big picture.
Is aspartate to oxaloacetate a transamination reaction as well?
Yes. Yeah, so she asked if aspartate to oxaloacetate is a transamination reaction as well, and it is. So we've got malate dehydrogenase working on the top, and we've got just a transamination reaction on the bottom. Any questions on that? Overall, pretty simple. We're really only dealing with like 3 different molecules. Okay, so a couple of the intricacies that come into play here is we've got alpha-ketoglutarate and glutamate. And we've got some like weird looking lines that's kind of hard to follow sometimes. So we're going to start on the inside. So we're going to start with glutamate. So this transamination reaction that turns oxaloacetate into aspartate is going to use glutamate in order, it's going to take something off of glutamate to make aspartate. So it's going to transaminate it, so it's probably going to put an amino group on there. Maybe not, though. What's different? Yeah, so yay. It's fun when you're right sometimes. So it's going to take off this amino group here. It's going to put it on to oxaloacetate to make oxaloacetate to make aspartate. And so when you remove that amino group from glutamate, it turns into alpha-ketoglutarate. That's what that's saying. So that's one of the products over here. So now aspartate comes over. We take off that amino group from aspartate, and we put it on this alpha-ketoglutarate, and it remakes glutamate, which can then go back across, repeat. This one comes back across, repeats. So it's kind of just reusing the same kind of molecules to help change all these different things into different forms. Did I lose y'all on that? Want me to go through it one more time?
Kind of just like recycling itself, sort of?
Exactly.
Okay.
Yeah, so why? Yeah, so really the only difference between all these, the only difference between aspartate, oxaloacetate is the amino group. So why make a whole new one when you could just transfer it over here, transfer it onto alpha-ketoglutarate to make glutamate, and then glutamate comes over here and says, hey, like you need this to get across, so I'm just going to give it back to you over here to go across. So this is why it's the malate alpha-ketoglutarate carrier, because malate can go through it and alpha-ketoglutarate can also go through it. Whereas this is the glutamate aspartate carrier because aspartate goes back to the outside, glutamate comes back into the inside. And those glutamate and alpha-ketoglutarate are just used for the transamination reactions. So something like, I think what he asked on the test was, I think it was saying, what do you, it was like, all right, so you've got oxaloacetate. What's going to happen? What's going to happen next? Something like that. So it's like, all right, you need to add glutamate to aspartate, so aspartate can then go through the shuttle. You kind of had to combine both of these into the same question. So really, if you saw glutamate being added to the equation, it would be down here, whereas if you're adding alpha-ketoglutarate into the equation over here to take off the amino group, you're going to be on this side. So just being able to orient yourself. Overall, what do we do? So overall, we took cytosolic NADH and we made it mitochondrial NADH. That was the whole purpose. Yes, because remember we produced those few NADH in the cytosol with glycolysis, yeah? But we can't make ATP with that there. We have to carry it to the mitochondria to go through oxidative decarboxylation. So this is how we get that NADH into the mitochondria. This is one of the ways. And we used, so pretty much y'all try ones, added NADH to your outfit to look like me. Because y'all like, because I know this, and so y'all added it, so y'all know it. But then y'all got rid of it once y'all got in the club, because y'all, you're not thinking about that. So that might have been the worst example ever. I'm sorry if that was. Questions? Do I need to go through it one more time?
No, that makes more sense.
I had one nod. Do we have any other nods? Okay, one more time. With feelings. Oh, y'all haven't had that class yet. Okay, so overall goal, cytosolic NADH to mitochondrial NADH. So we're going to take that NADH, add it to oxaloacetate. Makes what? Y'all are gonna fill in blanks. Makes malate. What enzyme?
Malate-dehydrogenase.
Yes, malate dehydrogenase. So malate, can it go through the membrane or no? Yes. Malate can. Yes. All right. So malate, we're inside. So
I have a question. If aspartate can go like out of the membrane, then why couldn't it just go back in? Or it does, or I don't know. You just like have to have the malate because it has the NADH.
The carriers are specific though, right?
yeah, so the carriers are specific to the molecules that they need. So yeah. So they're made specifically for malate because malate has that hydrogen on it now. So it's not going to let anything in unless it has a hydrogen because that's what's important. So hydrogen comes in, dumps the hydrogen, we're stuck with this. Well, now we need to recycle this back to the outside so it can make malate again. So it just turns it into something else to bring it outside, back to oxaloacetate, start over. So we're at malate. What's malate going to do?
Malate-dehydrogenase to oxaloacetate.
Yes, creating, most importantly, NADH. Yeah, so malate's going to go to oxaloacetate. In the process, removing NADH, we've accomplished our goal. We're in the club having fun. I guess y'all might, in theory, be the NADH because y'all get to go just stay in there. Whatever. I'll work on that next time. So you got oxaloacetate. So you're going to go turn into what next? Aspartate. With a what kind of reaction? Transamination. So what are we going to add to oxaloacetate to make aspartate? From where? Glutamate. So glutamate. So we're going to take the amino group from glutamate, creating aspartate, and alpha-ketoglutarate. So both of those are going to go through. All right, so now we're going to take that alpha-ketoglutarate. It's like, hey, give me my amino group back. So aspartate's like, sure, you can have it back. So now we've got glutamate and oxaloacetate again. Let me start over. And then really malate and glutamate can kind of both go through at the same time. Good? There's going to be at least one question, maybe 2 questions on the test on that shuttle specifically. It's not FADH2. It's only NADH. Because we didn't make any FADH2 in glycolysis, did we? Mm-mm. Okay, so if we want to go through the verbiage of it. So malate-aspartate shuttle, major producer of cytosolic or extra mitochondrial NADH is G3P dehydrogenase from glycolysis. Anaerobic oxidation, cytosolic NADH can't be accumulated since the reducing equivalents are constantly transported into the mitochondrial matrix for ATP via respiratory chain, whatever. Cytosolic NADH reduces oxaloacetate to malate in the cytosol. This reaction is catalyzed by what enzyme? Malate dehydrogenase. So it's also important it's the cytosolic malate dehydrogenase because we have a mitochondrial one as well. So this donates the pair of electrons to oxaloacetate, converting it to malate. Since the membrane is not permeable to oxaloacetate, it can't just go straight through, but malate can. So malate's now the carrier. So cytosolic malate is transported by malate alpha-ketoglutarate transporter. That's just the top one. Mitochondrial malate is reoxidized to oxaloacetate and NADH, most importantly the NADH, catalyzed by mitochondrial malate dehydrogenase. So there's NAD in the mitochondria. It takes the hydrogen, makes NADH. So thus, it can be said that cytosolic NADH is now transported and converted into mitochondrial NADH. And we know NADH makes how many molecules of ATP? 2.5, right? FADH, was that question on the test? No? Oh, well, you were ready for it. Excuse me. Oxaloacetate is then forms aspartate by a transamination reaction using glutamate. So it just takes the amino group off of glutamate. Aspartate goes through the glutamate aspartate transporter carrier. And then aspartate is then turned back into oxaloacetate via another transamination reaction using alpha-ketoglutarate to take back its amino group to make glutamate. Cool. Glycero-phosphate shuttle. All right. So I'm going to read this one first, actually. So this shuttle system, is it okay if I read it? Is it okay if I read it? Y'all can say no. Okay. The shuttle system transports reducing equivalents or pairs of electrons from cytosolic NADH to dihydroxyacetone phosphate. I'm just going to say DHAP from now on. So DHAP, cytosolic glyceraldehyde 3-phosphate dehydrogenase. We've already talked about that, right? That's the one that makes NADH in glycolysis. So the product is what? 1,3-bisphosphoglycerate and NADH. So cytosolic DHAP reacts with NADH and produces glycerol 3-phosphate or 3-phosphoglycerate. You got to know both of them. I don't know why you can't just pick one, but it's science. And NAD+. So going down here, this one's kind of difficult to follow, but we're starting with dihydroxyacetone phosphate. The whole point is to do what? Bring that cytosolic NADH and make it, and take that hydrogen into the mitochondria. So it's going to take that NADH using this enzyme, glycerol 3-phosphate dehydrogenase, to make glycerol 3-phosphate. So we're starting here, adding that enzyme, adding NADH, making glycerol 3-phosphate. This is super dry, I'm so sorry. I've got some water though, so it's good for me. So next. So glycerol 3-phosphate dehydrogenase is an NAD-dependent enzyme in the cytosol, whereas the mitochondrial enzyme form is flavor protein enzyme linked with FAD NADH2. What that's saying is we're turning this NADH into an FADH2 here. So that's like one of the few times where it actually changes. Actually, the only time that we're going to learn where it changes. We're making FADH2 because in this next page, when we're doing the electron transport, this complex 2 it uses FADH2. It removes the hydrogens off of FADH2 specifically to pump the protons in, which is important, right? We need to have one that does FADH2 because in TCA cycle we made FADH2, yeah? We made one of them. So I guess that one just gives it more of a job to do, so it doesn't feel left out. So we're going to take our glycerol 3-phosphate, and we're going to take off the hydrogen using this flavoprotein dehydrogenase. And that's going to turn FAD into FADH2, and that's going to turn glycerol 3-phosphate back into dihydroxyacetone phosphate. So that one's only a 2-step process where we turn DHAP into glycerol 3-phosphate or 3-phosphoglycerate using this enzyme and adding NADH. And then it takes the H's off and makes FADH2, turns it back into DHAP. Super simple. And then they'll go off and have their electrons stripped from them. So that's how we get the hydrogens into the mitochondrial matrix or the the matrix. Not that much. But if you don't study it, you're going to see all those words and be like, what the heck am I looking at right now? Like the faces y'all are all giving me right now. So next step, they go through the electron transport chain. So we're reoxidizing NADH and FADH2. O2 is reduced to H2O. That was the test question I missed on the test. So y'all don't miss it. Know what O2 is reduced to. I missed more than this one, more than that one on this test. So protons are expelled from the mitochondria, but free energy is stored in the resulting pH gradient synthesized. Free energy stored in the resulting pH gradient synthesizes ATP via oxidative phosphorylation. So we're using just like a muscle cell. We're kind of storing energy, potential energy, up in that outside the cell or outside of the inner mitochondrial membrane, and then we're going to use the passive, just going along the chemical gradient, the electrochemical gradient. That's going to help fuel the synthesis of ATP. So it doesn't take that much energy. Actually, it takes like very, very little energy. So it just takes a little bit to establish it, but the payoff is huge because it makes a lot of sense to where it can be very energy efficient to make energy. We'll get to it. So electron, it makes more sense when we actually get to it. So electrons are accepted by NAD, NADP, FAD, what have you. We've already done that. So now we've got our carriers. So we already know NADH is a carrier, FADH2, I guess technically would be a carrier as well. Now we've got something new, ubiquinone or coenzyme Q. So that's all it is doing. We can come over here. It just carries it from complex 1 and 2 and takes it to complex 3. So that's just taking hydrogens from those first ones and taking it to number 3. So, and cytochromes A, B, and C are just doing the same thing. They're taking hydrogens and transporting it further on as well. That's all they're doing. So our electron carriers. So they're arranged in order of increasing redox potential. Test question, potentially, if I got to see it. So increasing redox potential. What that means is, you know how when we did hemoglobin, where it had cooperative binding, I think was the word. So once it bound the first one, it was more likely to bind the second one of oxygen, the third molecule of oxygen, the fourth one, etc. It's kind of the same thing. So all right, we'll go to it. This first complex has the lowest redox potential. So, like, if anything, it's either going to happen or it's not. It's not really like that likely. But once this happens, this one's got a higher likelihood of happening than this one, and so on. So, increasing potential is what that means. I had to look it up earlier today. So the hydrogens or electrons flow through the chain in steps from the more electronegative components to the more electropositive oxygen. Got to know this order. Got to know this order. It shows up, I think, again here in a little bit. But got to know that it starts with NADH. This is going to be one of the ones where he's just going to put A, B, C, D, and it's going to be like NADH, cytochrome B, cytochrome C, then C1. He's going to have them all out of order. You got to be able to pick the right one. But I go NADH, Q, and then just remember, if you can remember, that it goes B, C1C, and A is at the end. So I don't know why it's not just ABC. Honestly, couldn't tell you. Those drunk Greeks, as Dr. G would say. All right, so got another order. We'll get to that on the next page. Oxidize is reducing equivalence. So really, it's just oxidizing the hydrogen off of NAD and off of NADH, off of NADH2, and putting it on to oxygen to make water. And there's my little note. I missed this. Don't be like me. So reviewing of what this is. So oxidation is we're losing the electrons. They're being oxidized. They're being taken off. So reduction is the gaining of electrons that's being reduced. So combined, it makes redox. What's the mnemonic, or what? Not the mnemonic. The oil rig. Yeah. What is that? Oxidation. Oxidation is lost, reduction is gained. Oil rig. Okay. So you think this chapter would be all about oxidative phosphorylation, but really it's just these last, it's really just this page, front and back. So everything else is just setting up this actual step. So we're ranged from lowest to highest redox potential. So we've got 4 different complexes. Ignore this for right now. We'll get to it. Don't worry about writing it all right now. But so electrons flow from complex 1 and 2. So complexes 1 and 2 go to complex 3. And coenzyme Q is what takes the hydrogens to complex 3. So complex 1 is going to take NADH, strip it of its hydrogen, making NAD+, and is going to transfer it onto cytochrome Q. Complex 2 is going to do pretty much the same thing, except with FADH2. It's going to take the hydrogens off, put it onto cytochrome Q, or coenzyme Q, and take it over here to 3. Then complex 3 is going to go to complex 4 via cytochrome C. So it's just going to take, for whatever reason, we need to go to the next one. So cytochrome C says, hey, they need to get there. I'll do the job. And at the bottom of the page, we've got our carriers again. So he's even listed it twice. So maybe just a little bit important.
Do we need to know where cytochrome B and C1 are, that they're in complex 3, or just know the general order?
I don't think he'll ask you where specifically those are at. He's got a couple other questions that he could ask you, which we'll go over here in a second. But Yeah, we'll just go through it. So we'll get back to that. So we've got TQs. So which complex oxidizes FADH2?
2.
And it's only 2. 2 is the only one that does it. And look, if we can see here, 2 is named succinate dehydrogenase. What did that enzyme do in TCA cycle?
FADH.
Yeah, so it deals with like FADH2. So like kind of cool how the body just reuses that. So now it would like double suck if you miss that one on test 3, just because. So definitely we need to know these names as well. There's only the 2 different names here. And I think he mislabels it on the next page, but we'll get to that after that. So you got to know which one which one deals with FADH2.
You mean the complex or the enzyme?
The complex. Then 2 is which doesn't have a iron sulfur cluster. So I remember I saw that on the test and I was like, holy, I didn't look at those tiny little details. So 1 does, 2 does, 3 does. This one's got a iron copper complex. So I guess that question probably should say which one has an iron copper in it.
Would it be 4 and 5?
Why is that technically complex?
Yeah, 5's weird. It doesn't really fit in the whole. That's its own whole thing. We'll get to on the next page. But definitely need to know that number 4 has the iron copper complex as opposed to the iron sulfur that the other 3 have. Don't know what those do. I'm guessing it helps take the hydrogens off or what have you. But it's different, so you need to know it. Then we also need to know which one doesn't shuttle, the hydrogens. So we can see here we've got some arrows going straight up. So complex 1 adds to the intramembranous space. 3 puts 4 hydrogens in. 4 puts a couple in. 2 doesn't. So 2 is kind of the oddball out of the group because it's FADH2 and it doesn't pump into establishing the gradient. Coenzyme Q takes what to where?
2 electrons from 1 and 2 to 3.
So it takes electrons from complex 1 and complex 2, takes them to complex 3. And cytochrome C takes what to where? Takes it from 3 to 4. I guess we'll probably zoom out, or I guess I'll stay zoomed in.
You say those again? Coenzyme Q takes what to it?
Coenzyme Q takes the electrons from complex 1 and 2 to complex 3. And then cytochrome C takes it from 3 to 4. So we just kind of like did the tiny little details of all this. Let's go through it overall. So complex 1, also known as NADH dehydrogenase. All right, because it's dealing with NADH, right? So that kind of makes a little sense. Complex 1 drives proton transport endergonically, right? It's requiring energy. We're going against the gradient. So it's going to be an endergonic reaction if it were. So energy is entering as opposed to exergonic. Energy is leaving from the matrix to the intermembrane space. So it's pumping electrons up there. So coenzyme Q oxidizes NADH. So now coenzyme Q is reduced. That should be a D there. Resuscitated. So now coenzyme Q, the reduced form, is the electron carrier, and it's going to take that electron to complex 3. So knowing the difference between oxidized coenzyme Q and reduced coenzyme Q, also a little important. But pretty We can figure it out. We're going to scroll down to the bottom here. Sorry, that wasn't on there. So complex 2 oxidizes FADH2, test question. So coenzyme Q does that. So now we've got reduced coenzyme Q. So this is where like it's different. So this does not drive proton transport into the intramembrane space. So go off the picture. And y'all can ask him about this too to double check it. But go off the picture because it says it does not drive proton transport. Or it says it does, but it does not. And then so coenzyme Q, and transports it to number 3. So it does the same thing except with FADH2 instead of NADH, and it doesn't drive proton transport. It does not. So go off the picture on the previous page. So now we're at complex 3, and we've got reduced coenzyme Q. I'm sorry, these are like super weird. I don't know why it's all like code word, what have you. But so number 3 is going to oxidize coenzyme Q. So it's going to take those electrons, right? It's going to oxidize coenzyme Q, and it's going to put it on cytochrome C. So now coenzyme Q is oxidized, cytochrome C is reduced. In the process, it's going to drive protons into the space. And that's really all it does. That's it. Yes. Yes. So now we get to 4. So 4, cytochrome C gets to 4, and it is going to be oxidized. So this is kind of like the last step in the oxidation process. It's going to lose those electrons, and it's also going to drive proton transport. But also, 4 does something important too here that we didn't talk about. So I probably should add this to my TQs. So we can see that 4 takes oxygen, electrons, and hydrogens and makes water. So complex 4 turns oxygen into water. So that goes all the way back to this equation over here where we started with glucose, added oxygen. We made our CO2 in TCA cycle and our water. So this is where this water is finally being made from that full equation. But it's not directly being made, right? That didn't directly happen. It was an indirect process. Talked about that like 20 minutes ago. So got to know that. And we still haven't made energy yet. We've taken all this time. It's just like the biggest tease. We haven't done anything. But we're getting there. So now we're at complex 4. Complex 4 is done. We've made our water. We've made the gradient. Now it's time. We're just going to skip ahead just a little bit. And it's time to make our ATP. So this picture, so we've got number 5. I guess we've got 1, 2, 3, 4, and now we've got number 5 over here. It's really made up of 2 different kind of subunits. So we've got subunit F0 and subunit FO, F1 ATPase, also known as proton ATPase, also known as H plus ATPase, also known as ATP synthase. So really, if you see anything else other than just the FO, it's the second one. That makes it a little bit more simple. I think he likes this one, the FO, F1 ATPase, or is that a 1, or I can't tell. So what we've got is, so the FO subunit is really just allowing hydrogens to come from this Oh, I didn't talk about this before. So we've got the intramembranous spot, the P side and the N side. Wherever the N, where's N side? P just stands for positive, N is negative. So P is just there's more positive because there's all those H plus ions, right? And then N is just relatively more negative because it doesn't have so many H plus ions on that side. So the F0 subunit is going to allow those hydrogens just to flow along the gradient towards the negative side. So it's going to go from positive to negative and going through here. Well, the FO, F1 ATP synthase, it's like a generator. So what's happening is that it's spinning, and it's going to add an ADP and a phosphate. And so these hydrogens that come down, it's going to get like a turbine combined and make ATP. They don't know exactly how it works yet, but moral of story is ATP is finally made.
Like our class doesn't, or like science in general?
They don't know like 100% exactly how that works yet. They know that like it's divided into the two subunits. Hydrogen flows down here. It's like a turbine. It just, I guess like the kinetic energy is what allows it to combine the phosphates, to combine this third phosphate to ADP, making ATP. But they don't know quite 100% how yet. So we've got energy. We made it. We did it. Yeah, and there's much rejoicing. A little anticlimactic. I could probably build that up better next time. Next time, we all try to. But that's it. So we've gone through the entire oxidative phosphorylation. But I've got 5 more minutes with y'all. Just because I'm mean, I'm not. I'm actually helping. So going back to this previous page, blockers of electron transport chains. So something that's regulating. Do we have a lot of regulation questions on the previous exam? Yeah?
I believe so.
So we had at least a few. So definitely need to know these 3 examples here. So retinone blocks the electron transport from NADH to coenzyme Q. So what complex is that? 1, 2, 3, or 4?
1.
I heard 1, 1 and 2.
NADH602 is 1.
It's just 1, right? Because complex 2 is FADH2, right? So retinone does that. So my little remembering thing here is retinone doesn't get past step 1. So retin 1 doesn't get past step 1. So it blocks it really early on. So then we've got antimyosin A blocks the electron transport from cytochrome B to cytochrome C1. We didn't really necessarily talk about those, but those are in, let's see which one of those in. B to C1 is in complex 3, yeah? So that's going to be in complex 3. I have a process of elimination for that remembering one. So next we have cyanide and carbon monoxide block the transport from A and A3 to oxygen. So carbon monoxide or CO can't make O2. So if y'all have ever heard like carbon monoxide poisoning in your house, right? It's like the silent killer, I'm pretty sure. So if you're breathing that as opposed to breathing O2, you're not going to make energy and you're going to die. So yes, you will die, and it will be very sad. Unless people hated you, then they'll be rejoicing. So and then so elimination, cytochrome A is between B and C1. Got to know those. He's going to put at least one of those on there. And over here on this next page, the third one blocks going from A and A3 to making O2. And that's with carbon monoxide or cyanide. Right? So people, what was it like? People used to have cyanide in their teeth, right? they're like the spies. They like crack it open, right? So exactly. So really bad. So really important that we don't have cyanide or carbon monoxide in our bodies because we will die. It will not be fun. So click over here. Passage of 3 hydrogens is required to synthesize 1. Passage of 3 hydrogens is required to synthesize 1 ATP. You might ask it, kind of similar along the lines of NADH, FADH2. Y'all can read this yourselves. Just get y'all out of here. Yes, I'm from Texas. I say y'all's selves. Lastly, uncouplers of oxidative dephosphorylation. So we were regulated on this side. So this is in the transport chain, kind of with our carriers, but we can actually block the ATP synthase as well. So 2,4-dinitrophenyl and FCCP, I'm not going to say that word, uncouple between the electron transport chain, so the TCA cycle, and oxidative phosphorylation. So they can stop ATP biosynthesis, but they don't interrupt the electron flow through the chain. So really, it just cuts off that last step. So it goes through all this, and then it just stops that last part. It does not make ATP. So got to know 2,4-dinitrophenyl, FCCP. And also down here, brown fat. Who has brown fat? Babies, right? All the girls said that. No guys. I wonder why. All right, so brown fat in the newborn tissues contains large numbers of mitochondria and has high cytochrome levels. It oxidizes fuel, but it does not produce ATP because mitochondria does not couple the electron flow with oxidative phosphorylation. So babies have a special protein, thermogenin. Maybe know that protein, thermogenin. He bolded it for you in the middle of a large paragraph. And that's what uncouples it. So because they have enough, that's just what their brown fat does. They need to keep warm. So it helps dissipate heat and maintain body temperature. Brown fat, you learned something. Go home, sleep. Sorry to torture y'all an hour after multiple tests. Questions?